All right, it's my pleasure to introduce Courtney Harrison. She earned a BS in Animal and Nutritional Sciences from West Virginia University in 2018, and an MS in Fisheries and Wildlife with a specialization in Wildlife Disease Ecology and Conservation Medicine from Michigan State University in 2021. With a growing interest in fish population health and a passion for studying infectious disease in fish, that led her to pursue a PhD at Auburn University under the guidance of Dr. Tim Bruce, where she is working currently on the development of Flavorbacterium vaccines. So take it away, Courtney.
Thank you, Nick. Can everyone hear me okay?
Yeah, sure can.
Awesome, thank you. Yeah, first I want to thank the organizing committee for hosting this event. Again, this is my, I think, third time presenting to the American Fisheries Society Fish Health section, so I'm happy to be here. I'd also like to acknowledge the PIs on this collaborative project. And so today I'm going to talk to you about some current and prospective work that I'm doing here at Auburn University that involves identifying candidates for a live attenuated vaccine against Colinaris disease. So the catfish aquaculture industry is the largest aquaculture industry in the United States. In 2021, it reached $398 million in total sales. The top three catfish producing states that comprise 97% of total catfish sales includes Mississippi, that produced approximately 93 million pounds of catfish in 2022. And that's followed by Alabama and Arkansas. But unfortunately, a critical threat to this industry is disease, which is caused by bacteria, viruses, and parasites. So bacterial diseases can cause up to 60% of catfish losses. And the top three bacterial pathogens that producers are grappling with are virulent Aeromonas hydrophila, Flavorbacterium columnar, and Edwardsella ictalori. And so catfish production is a little different from other aquaculture fish species. So it's much more extensive. Producers have limited control on temperature and predation to birds. It's, an open system, so there's a lot to deal with. So whenever a disease outbreak does occur, it's very difficult sometimes to mitigate this outbreak. So this can mortality gain. This will increase the cost in treatment as well. For this talk, I'm going to focus on one particular bacteria called Columnar. It belongs to the family over the years. So it was formerly referred as Bacillus columnaris, and then eventually Flavorbacterium columnaris as Flavorbacterium. And by being negative, pigmented, and having a liability. And of course, this causes the infamous Columnar.
We lost Courtney here. Hopefully she jumps back on quickly. Just hang tight, folks. Or Matt, if you have any jokes for us.
Well, no, it's not. I'm just glad it wasn't me.
Oh, you're back, Courtney. Let's try this again. You might want to.
I'm so
Hey,
sorry
Courtney.
about that.
Hey.
All right.
Was that okay? All right. I'm going to pick up where I left off.
Yeah.
So I think it was right around here. So.
You're still aren't sharing yet.
Oh, I'm still not sharing. Oh, okay. Hang on just a second. Apologies.
Oh, good.
Okay, can you see my screen now?
Yep, and you sound good.
Okay, awesome, thanks. All right, so Flavorbacterium columnar, it causes Colinaris disease. And this disease in particular can lead up to $30 million in economic losses per year. So it's a very serious bacterial pathogen. So some clinical signs of Colinaris disease include necrotic gill lesions, like in this picture you're seeing here, which can sometimes be white or yellow, and that's from a massive amount of bacteria collected at this site. And if you take these gill lamellae and put them under a microscope, another telltale sign of Colinaris disease is the presence of these filamentous haystacks. And then another sign are depigmented skin lesions like the one here on this catfish. This is the saddleback lesion. Also, the presence of frayed fins. So some treatment and prevention options for Colinaris disease include salt bath treatments, chemical treatments such as copper sulfate and potassium permanganate. But I should note that even though these chemical treatments are used in the field, they're officially not approved. There's also antimicrobials such as Aquaflor and chloramine T, but the most desired among these treatment options is vaccination. So there is a vaccine that's licensed for use in channel catfish and largemouth bass called Aquavac Col. However, in under production settings, it's not efficacious, and so it's no longer being sold. And so one potential reason for the lack of efficacy is suboptimal host pathogen dynamics. So the genetic diversity of Flavorbacterium columnar has been studied extensively. In 1998, a study released that strains of Flavorbacterium columnar were morphologically similar, yet they were biochemically different. And then in 1999, 3 genovars were proposed for the species based on phenotypic and biochemical similarities. However, these strains were genetically different. And then in 2017, a study released that proposed 3 or 4 genetic groups based on comparative genome analysis. And then in 2018, the species was officially categorized into 4 genetic groups to more accurately demonstrate its genetic diversity. And there were also biological associations between the genetic groups and host species. So for example, genetic group 2 is mainly associated with catfish aquaculture, whereas genetic group 4 is mainly associated with Colinaris disease and tilapia. And so earlier this year, LaFrance et al. Published an extensive genetic characterization and revealed that Flavorbacterium columnar now represents 4 distinct species, including Flavorbacterium columnar, Flavorbacterium covei, Flavorbacterium davisi, and Flavorbacterium aureochromis. So from this point forward, they are collectively known as Colinaris causing bacteria. And so regarding the efficacy of the vaccine I mentioned earlier, it's important to note that that live attenuated vaccine was created using Flavorbacterium columnar, or a genetic group 1 isolate. So with this newfound knowledge of Colinaris causing bacteria, causing species diversity, and having an affinity for different host species, vaccine development has been reinitiated. So this project's hypothesis is the development of a live attenuated vaccine by targeting virulent Colinaris causing bacteria with specific host affinity could provide protection in catfish and other fish species against Colinaris disease. And so to address this hypothesis, this project has 3 main objectives. So one is to develop and identify vaccine candidates to control Colinaris disease in farm-raised catfish and other cultured fish species such as tilapia, baitfish, and rainbow trout. Number 2 is to test the efficacy of this vaccine and identify the optimal delivery method. And objective number 3 is to develop and test this vaccine under experimental pond conditions. And so, as I mentioned earlier, this project is collaborative. So the work has been divided among institutions involving fish health specialists with many years of experience working with Colinaris causing bacteria. So here at Auburn University and the USDA Auburn unit will be focusing on isolates associated with catfish and tilapia. Michigan State will focus on isolates associated with baitfish. The University of California, Davis will focus on rainbow trout. And at Mississippi State University, that's where we'll do experimental pond trials and catfish fingerlings. So now I'm going to go into more detail on objective 1 for this project. So the method of attenuation that we've chosen is a rifampicin attenuation strategy. So rifampicin is a broad spectrum antibiotic. It inhibits the DNA-dependent RNA polymerase. And it's been used before to create a mutant attenuated strain of Brucella abortus. So this strain was used to immunize mice against a virulent strain of Brucella abortus. And that was later turned into a live attenuated vaccine that's still licensed today for use in cattle. And so this attenuation strategy has also been used in aquaculture settings. So in 2008, a Flavorbacterium cycrophyllum strain was used to vaccinate rainbow trout via immersion, and the fish exhibited protective immune responses. And then in 2013, a live attenuated vaccine against Colinaris disease was used. So a rifampicin-resistant Colinaris causing bacteria provided protection in channel catfish and tilapia. And then in 2015, a live attenuated vaccine for enteric septicemia of catfish was used in a form of a vaccine feed mixture, and catfish fingerlings were immunized under a production setting, and this provided protection to the catfish fingerling when they were challenged with the wild type strain. So for this project, we'll be using the same strategy. So I'll take virulent Colinaris causing bacteria, revive them from cryostock onto modified shea agar, and after 48 hours of growth, at 28 degrees Celsius, I'll then start increasing I'll then pass them to a plate with increasing concentrations of rifampicin. And so to confirm the attenuation, I'll do this by in vivo immersion challenges. So this involves multiple rifampicin resistant mutants. So this includes the mutant strain, the parent strain, and a control in triplicate with 20 fish per tank. I'll do this by immersion for 30 minutes in a flow-through system. And at the end of this experiment, I'll calculate the cumulative % mortality. And so another goal of objective 1 is to determine the mechanisms of attenuation. And so I'll do this with a series of analyses, starting with whole genome sequencing, followed by a protein profile of the attenuated isolates and their wild type parent strain via SDS page. And then I'll use a Western blot to look for antigenic regions of interest. Okay, so now I'll go into more detail of objective 2. So using the vaccine candidate obtained from objective 1, I'll use this to immerse I'll use this to identify the optimal vaccine, the optimal method of delivery for this vaccine. So the methods of delivery that I'll test are immersion and oral delivery. So both of these have their pros and cons. Immersion, this can be done in smaller fish, usually before stocking into ponds. However, this is sometimes stressful to fish, and because the fish are smaller, their immune defenses are not fully developed. Whereas in oral delivery, this can be given to fish in ponds. It's a little more practical under production settings. The fish are a bit larger. However, there's a history of oral vaccines being weaker or providing a shorter period of protection. But regardless of their pros and cons, we're going to figure out which method of delivery is optimal for this vaccine. And so for vaccine optimization, I'll test multiple parameters, including dose, exposure time to determine the level of protection via antibody titers. And then I'll compare the efficacy of the vaccine in different fish sizes. So now for objective 3, we'll take this optimized vaccine and put it to use out into the field, so in fish and ponds. So this will be done at Mississippi State University at NWAC. So we'll take that optimized vaccine obtained from objectives 1 and 2, and I'll immunize catfish fingerlings under a standard commercial production cycle for catfish fingerlings. And so approximately 28 days after they're vaccinated, I'll challenge them with the wild type parent strain. And then at the end of this study, I'll calculate cumulative % mortality, relative % survival, and food conversion ratio to determine the efficacy of the vaccine. And so here you're seeing a project timetable. So the project is already underway. It officially started in January of this year. And so currently I'm still working on attenuating strains of Colinaris causing bacteria. And most recently I began confirming some of the attenuated isolates in catfish. So some project outcomes include an optimized or optimization of a novel vaccine for aquaculture fish species. We hope to produce a viable vaccine candidate against Colinaris disease. And then a long-term goal of this project is to reduce losses in economical fish species within the southern region. So at this time, I'd like to acknowledge USDA NIFA, the Southern Regional Aquaculture Center, for funding this project, USDA ARS here in Auburn for providing tilapia and catfish for this study, Dr. Ben LaFrance for his research collaborations. I'd also like to thank Dr. Matt Griffin, Dr. Esteban Soto, and Dr. Thomas Locke for their research collaboration and serving as principal investigators on this project. I'd also like to thank my colleagues at the Bruce Health Fish Laboratory. And then last but not least, my primary academic advisor, Dr. Timothy Bruce, and my secondary academic advisor, Dr. Eric Peatman. And I will take any questions at this time. Thank you.
Great, thank you very much, Courtney. Excellent presentation.
Thank you.
Folks, feel free to unmute yourself and ask Courtney a question directly, or you can drop it in the chat.
Hey, Courtney. Great talk.
Thank you.
Are you attenuating strains just from FCOV? Are you using the 4 different species? What isolates are you actually proceeding with to make these attenuated candidates to test?
Thank you. So maybe I wasn't very clear, but at Auburn, we're focusing on, so I'll be focusing on attenuating genetic group 2, so FCOV, genetic group 3, which is F. davisi. That has been, some of these are also known to be virulent in tilapia. So at Auburn, we're focusing on catfish and tilapia. And then genetic group 4, which is Flavorbacterium aureochromis. So there's 3 different strains that I'm currently attenuating for this vaccine.
Okay, so 1 strain from each of those 3 species.
Multiple strains.
Multiple strains.
Yes.
Okay, that was what I was curious about.
Yes.
Cool.
Thank you.
Yeah, Tom, I see your hand raised.
Hi, Courtney. Great
Hi, Tom.
presentation as always. So I'm wondering if you'd be willing to share just a glimpse of what you've been finding, because you mentioned you've been doing some of the pilots with the attenuated strains. So if you're willing, maybe sharing how the attenuation is going in catfish.
Sure. Thanks for asking. So I've attenuated about three strains in total now. 2 of them being in tilapia, 1 in catfish, and I've had the opportunity to test the attenuation in the immersion challenges that I talked about. And I found that that the highest concentration of rifampicin resistant isolate so far, I got 0% mortality in an 8-day study. So it's very encouraging and exciting. But yeah, so far just one isolate I've attenuated successfully now, but we're going to test it again just to make sure. And then the next step is vaccine challenges. So I'll actually immunize catfish and see if it provides protection.
Very cool. Thank you.
Thanks.
Courtney, you got a question in the chat. You may have just answered it though, just to be sure. Are there any differences among strains during attenuation?
Are there any differences among strains in attenuation? Yes, I have found that some are a little easier to attenuate than others. It's a long process. So for anyone who has worked with attenuating bacterial strains, they're all different. It's never textbook, and like some, again, like attenuate quicker than others. But I'm having fun doing it. I'm learning a lot. So yeah, but between, you know, the different strains of Colinaris causing bacteria, F. covei, F. aureochromis, they're all unique, is what I'll say.
Matt?
Hey, Courtney, now that you've dug into this a little bit and you started, do you have any recommendations or strategies for other folks that are kind of taking on a similar task? You know, as far as smaller leaps, larger leaps, frequent freeze backs, etc. What would you recommend to someone that's just starting out with a similar project?
Oh yeah, that's a great question. Thanks for asking. I save back all of my attenuated isolates. So I started small. So I started with a concentration of 0.01 micrograms per milliliter of rifampicin. Then I raised that up to 0.5 and then 1.0 and 5, 10, 20.
Are you frozen too, Nick? Oh, okay. Shucks.
There was a great lesson learned she was about to say. So Courtney, if you can hear us, you're frozen. We'll give her just a couple seconds here and then we'll.
When the IT issues reign, they pour.
Yeah.